Hi there. In the previous video, we studied meiosis and saw that it was responsible for the production of gamete cells, which lay at the foundation of sexual reproduction and inheritance. In this video, we're going to look at the original studies of inheritance performed by Gregor Mendel in the late 19th century. We'll see that he studied the inheritance of 7 traits of pea plants, that he carefully bred plants with pure attributes and quantified the outcomes, and we'll see that 2 genes can be inherited independently even if they're on the same chromosome due to crossing over. On this slide, we're introduced to a lot of important terms for the discussion of inheritance. The 2 most important are perhaps genotype and phenotype. Genotype refers to an organism's genome and all the genetic variation that it possesses. Phenotype is physical characteristics, whether that be, in this case, wild type or a mutant fish. Now, in this example, wild type fish looks somewhat normal, and the mutant fish has this bent characteristic. This slide is suggesting that there is a gene A for which comes in 2 alleles. Alleles is a term for alternative forms of a single gene. It's represented by dark black for the dominant wild type and the striped pattern for the recessive mutant form. Now, if an organism has 2 copies of the wild type dominant gene, large A, it's going to have the normal phenotype. So if the genotype is big A, the phenotype is normal. Since big A is dominant over little a, if an organism is heterozygous for this gene, meaning it has 1 dominant copy and 1 recessive copy, it's still going to have the wild type phenotype because it appears normal. It's only when the fish has 2 copies of the recessive gene that it's going to be homozygous recessive with copies little a, and then it displays the mutant phenotype. The last important term that I have to mention is locus. When we talk about a locus on a chromosome, that refers to a space where a gene is located, and you can talk about genetic variation at different loci in the genome. But the key point here is that the genes you inherit, your genotype, will influence your phenotype, and that's the foundation for Mendel studies. Mendel chose to study 7 characteristics of pea plants, which happen to have a lot of variation. He studied, and those 7 are listed here on the top. Seed shape could be round or wrinkled. Seed color could be yellow or green. The flower color could be purple or white. The flower position could be axial or terminal. The pod shape could be inflated or pinched. The pod color green or yellow. And the plant height could be tall or short. So these are 7 traits. It turns out that for each of these 7 traits, there is only these 2 options. For instance, the flowers are either pure purple or pure white. You never see sort of a light pink-ish flower that represents a blending of the 2 the phenotypic traits. A second important fact, at least retrospectively for his studies, is that it turns out that each of these properties are determined by a single gene. That's not always the case. Genes that contribute to your height, for instance, there are hundreds of genes that influence your height. And that's one of the contributing factors why we have such a smooth, wide range of distribution of heights. But using as a height as an example, the plants are either tall or short. They were not a wide variation or a bell-shaped curve. A third important trait for his studies is that all traits are completely independent. There's no correlation between traits. So, for instance, it's not the case that purple flowers tend to have yellow peas. Purple flowers are equally likely as white flowers to have either yellow or green peas. And lastly, I'll mention sort of what some people view as a lucky accident. Others might say there might be more to the story than we know, but Mendel happened to choose, it turns out peas have 7 chromosomes, and Mendel happened to choose these 7 traits, and each one of these traits is inherited on one of the different 7 chromosomes. Quite a coincidence at the very least. But as we'll see later in the lecture, that doesn't necessarily matter. Genes that occur on the same chromosome can still be inherited independently. However, if it's the case that 2 genes lie on the same chromosome and are very physically close to each other, then traits like this can be correlated, that you can definitely find that something like purple flowers might be much more likely to have yellow or green peas. But that's not the case in Mendel studies in any way. Now, we've seen this slide before, so I won't spend much time on it. Again, in this case, there were 3 different types of chromosomes, and therefore, since the chromosomes are inherited independently, there are 2 to the 3rd power possible combinations, and this is the number of combinations for chromosomes being inherited completely as a whole unit, which we know doesn't happen. Because of crossing over, we have essentially infinite possible number of unique genetic inheritances. Each chromosome can cross over at a different base, and so there are many, many possibilities. And just to make the crossing over story a bit more concrete, with different genes being listed as opposed to just colors, that if you were going to inherit either a full copy of your maternal or paternal chromosome, the only 2 possibilities would either be large A and large B or little a and little b. However, because of crossing over, you can have unique combinations where this material is crossed over and someone can inherit a large A but then a recessive version of gene B. The farther the distance between A and B, the more likely that a crossover event is likely to occur between them. If the 2 genes are very close, then it is unlikely that a crossover event will happen. And that point is made even a bit more explicitly here, highlighting 2 genetic loci, 2 genes, gene A and B, which are physically very close on the chromosome, and there's unlikely to be a crossover event, so almost never would you expect to see the genotype big A, little b, or vice versa. You're always going to see big A, big B, or A and B. If A and B are never independently assorted, in modern parlance, we will talk about that this represents a haplotype, which essentially means a block of chromosome that's always inherited as a unit. However, when you consider this haplotype of big A and big B, the other genetic loci are at a great enough distance away that you're just as likely to inherit large C or little c, large d or little d, so that you will get independent assortment then for any of these genes with this haplotype block. So here are the results from one of Mendel's breeding experiments. He took yellow-seeded plants and green-seeded plants and manually cross-fertilized those. So this is known as the parental generation, referred to as the P1 generation. The first set of offspring are called the F1 generation, and 100% of the offspring from this cross is yellow-seeded plants. And that's because yellow color is dominant to green for the color of the seeds. However, when you took 2 F1 peas and crossed them, then you produce the F2 generation. And here you see 75% yellow pea plants and 25% green plants. These results show 2 major facts. The first one being, which we take for granted now, is that the gene for the green seeded pea plants is not lost in the F1 generation. It's there, it's just recessive. And secondly, when he crossed this yellow F1 with another yellow F1, you obtain both yellow and green plants, so that's clearly not a result of blended inheritance. It points to the fact that the color only comes in 2 varieties here, based on the genotype of the individual seeds. So here's how you can explain the result or calculate or predict what the result would be for a breeding experiment. The first step you need to do is when you're given a phenotype, you have to have some information about the genotype. You know, since we've mentioned that green is the recessive color, that the only way you're going to get a green plant is if you have 2 copies of the recessive allele, so little y, little y. I'll also say that the letter chosen for a gene is completely arbitrary. In this case, you could be using big T and little t. It's just somewhat convenient to use Y for the dominance because the dominant color happens to be yellow. But when we know that we're breeding it with a yellow pea, there's 2 different ways we can have yellow peas, right? It turns out, since these are purebred plants, that they are homozygous for the dominant gene, so they could be YY. You have to at least consider, unless you're given extra information though, knowing what you know about seed color, that this original experiment could also contain peas that are big Y, little y, that are heterozygous. So when you're asked to do a genetic cross or predict the results of one, you'll have to look for the clues that will tell you what the genotype is given a phenotype. So since this is something you'll need to be able to do, I'll go through this in a little bit more detail. After a cross, you need to report the genotype and the phenotype of a generation that you're being asked about. In this case, for the F1 generation, the phenotype is yellow seeds, the genotype is heterozygous, big Y, little y. But now we look at the production of the F2 generation. Well, since we're going to cross a YY with a YY, and it's just like a matrix multiplication. We write out the chromosomes here for the egg and the sperm on 2 axes, and we do the multiplication, if you will, and we compute the genotypes at any particular loci. And we have 4 different possibilities, and the results are 75% will be yellow, 25% will be green. The genotypes, however, are 25% big Y, 50% big Y, little y, sorry about that line, and 25% little y. So if you're reporting the genotype and phenotype here, you say phenotype is 75% yellow, 25% green, but the genotypes are a ratio of 1 to 2 to 1, big Y to big Y, little y to little y. And this simple method of calculating genetic traits can be generalized to predict more complex cases where you take, in this case, a yellow smooth pea with a green wrinkled pea. Again, you need to have some information to deduce what the genotype is, and then you can do the crosses. The only gamete that can be formed from this plant is a large Y, large R, and the only type of gamete that can be produced from this pea is little y, little r. Now we're going to cross those. We're going to get every plant is going to be big Y, little y, big R, little r. So everything is going to be yellow and round. But now on the next slide, we're going to consider the production of the F2s. So we're going to do a cross with the F1. So we're going to take 2 peas that are each big Y, little y, big R, little r, and then we'll do the calculation. And so here that is. And I'll just write out again the genotype for the peas that are producing the gametes. As you can see, there are 4 different types of gametes that can be produced by such a plant. You can have big Y, big R, you can have big Y, little r, you can have little y, big R, and you can have little y, little r. So there's 4 different possibilities. And then those are written out along these 2 axes, and you do the multiplication. Now there are a lot of things that you can notice here. First, let's only look at color. You can see that just like in the previous experiment, if you consider 4 as a unit, it is a ratio of 3 to 1, yellow to green, just as in the case where we're only considering a single gene. Likewise, if we look at smooth and wrinkled, we have 4 wrinkled P's. We have 12 possible smooth P's. So once again, you have a ratio of 3 to 1 on smooth versus wrinkled. If you look at the phenotype, 9 out of the 16 possibilities are yellow and round, 3 out of 16 are green and round, 3 out of 16 are yellow and wrinkled, and only 1 out of 16 are green and wrinkled, are recessive for both traits. However, if you ask a different question, what is the most common genotype, you'll see that there are 4 examples of big Y, big R, little r right here in the center. So the 4 different genotypes produce a single phenotypic result. There is 1 type of big Y, big R, big R. There are 2 of these combinations, and then there are 4 of these. So that's a total of 9 out of 16 possibilities. We'll practice this type of analysis in class, and you will be prepared to do this also on your own for the test. Okay, thank you very much for listening.